Hi, everybody. This is Mr. Mittner. We have a test tomorrow, and I'm hoping that this will help you get ready. We have macromolecules was the area that you said that you needed the most help on. Most people said they were okay with pH, okay with enzymes, but we needed a little more help with the macromolecules. So I made this mind map, and mind maps are an incredible way to see things. Some people like to do outlines. Other people don't see things in that linear flow. They need to see the things spread out. So this is a mind map. In the middle of the mind map here, we have macromolecules. And it goes off into 4 different areas. So we're gonna look at this. The first one we're gonna look at is carbohydrates. Carbohydrates are made of monosaccharides that form polysaccharides. A typical monosaccharide would be like glucose or fructose. That's a single G, that's a single sugar. And then they join together to make disaccharides and then polysaccharides. Polysaccharides are either used for storage usually, and for storage in plants it's starch. That's the polysaccharide where monosaccharides join together in plants. And for storage of energy in animals, it's glycogen, and glycogen stored in the muscle and liver cells. Also, polysaccharides can be used for support in arthropods and animals is chitin. That's that crunch when you break a crab leg and you step on an insect. Or cellulose in plants in the cell walls. They provide structure and support. And we'll talk more about cell walls later on next week. Carbohydrates are made of CHO, carbon, hydrogen, and oxygen, and a formula of CH2O, meaning that carbon, water, carbohydrate. So carbon and oxygen are going to be relatively close to each other. Every time one of those monosaccharides joins with another one, there's a water given off through something called dehydration synthesis. So the oxygens and the carbons won't be exact, but they'll be fairly close. Much, much more closer than the other one that's CHO, and that's lipids, and that's where we're going to go next. Lipids are down here. There's 3 types. There's waxes, steroids, and fats. Under steroids, I gave you 3 types. There's cholesterol. And in cholesterol, there's LDL and HDL. Remember, HDL is the good type that cleans out your arteries and stuff and removes the bad stuff from the walls of your arteries. LDL forms plaque and causes dangerous situations where you have a higher incidence of heart disease and possibilities of stroke when you have really high LDL in your cholesterol. Other 2 types of steroids are testosterone for males and estrogen for females. The last type of lipid is fats, and we broke those into saturated and non-saturated or unsaturated fats. Saturated fats hold the most hydrogen possible because they have no double bonds on those carbons. You got a glycerol backbone with 3 tails, glycerol tails, glycerol backbone with 3 fatty acid tails, and those fatty acids have as many hydrogens because there's no double bonds. So there's no kinking. Because of that, it's a solid at room temperature, and these usually come from animals. The only exception to that is like coconut oil. Unsaturated fats do not hold the maximum number of hydrogens because they have double bonds. They're kinked up, and so you get, they don't get as close together, so you got liquids at room temperature, and they usually come from plants. All lipids are made of CHO, carbon, hydrogen, and oxygen. Much less oxygen than what you found up here in the carbs. And lipids are nonpolar, which means they're not soluble in water. They want to move away from water as much as possible. The last 2 macromolecules, we have nucleic acids. Their base is, their monomer is the nucleotide. A nucleotide is made of a sugar, a phosphate group, and a base. And those nucleotides join together to make nucleic acids. They're made of CHOP, carbon, hydrogen, oxygen, nitrogen, and phosphorus. The only one that has a lot of phosphorus in it. So if you see a lot of phosphorus in the formula, it's most likely a nucleic acid. And the 2 types of nucleic acids are DNA and RNA. These are used for genetic storage. All the information about your genes and where you get your information, for what you got your eye color and your hair color and all that stuff from your mom and dad is in your nucleic acids. We're going to spend an entire 9 weeks on nucleic acids. From Christmas to spring break, we're going to talk about this in great detail. And then the last one is proteins, and we're going to talk about these all year long because they show up constantly. They have many, many jobs, including one that we've already talked about, and that's enzymes. Enzymes are proteins. All proteins, the monomer of that, are made of amino acids. There's 20 different types, and they bond together. And proteins are made of CHON, carbon, hydrogen, oxygen, and nitrogen. Proteins do so many things, and it's all based on their shape. Remember, their shape depends on that amino acid sequence, because they'll bend and they'll twist, and the polar molecules want to be close to the polar molecules, and the nonpolar molecules want to be close to the nonpolar molecules, but the polar and the nonpolar don't want to touch each other, and so they'll move away from each other as they move towards the other polars. They'll move away from the nonpolars. It's crazy, but that shape makes it do what it does. Remember, enzymes have a specific shape. That substrate has to bond into it in that active site, and if that shape is screwed up, the enzyme doesn't work. I hope this helps. These are your macromolecules. You also have pH. Remember, in pH, 0 is low and 14 is high and 7 is neutral. Anything below 7 is an acid. Anything above 7 is a base. It's a base 10 scale, so every time you move one number, it becomes more acidic by a power of 10, or more basic by a power of 10. More acidic has more hydrogen ions. More basic has more OH, or hydroxide ions. And then the enzyme substrate, active site, temperature, salinity, and pH all affect enzyme behavior, and we'll get that down when we get to those labs that we didn't get to do last week. So I hope this helps. Good luck on the test. Study hard, and I will see you tomorrow. Thanks. Have a good day.